Hello and good afternoon, everyone. Thanks. I want to thank the organizers for including me in this series. My name is Rune Stensvold. I hold a PhD and I'm a senior scientist at the Laboratory of Parasitology, Department of Bacteria, Parasites and Fungi at the State and Sim Institute in Copenhagen in Denmark. And I'm a parasitologist, and my sort of entry to infectious keratitis was through acanthamoeba, which is a rare but quite important cause of infectious keratitis, and which, as you know, it can be acanthamoebiasis can be a quite sort of debilitating disease of the cornea. So the title of this presentation is Detection and Identification of Acanthamoeba and Other Non-Viral Causes of Infectious Keratitis and Corneal Scrapings by Real-Time PCR and by Next-Generation Sequencing-Based 16S and 18S Gene Analysis. So, as you know, infectious keratitis can be due to a number of different microorganisms, including parasites, bacteria, fungi, and viruses. And here at the SSI, we have a metabarcoding platform in place, which is also referred to as the microbiome platform. It's amplicon-based, and it was developed in our lab and relies on the use of 1' pair for 16S and 3' pairs for 18S ribosomal genes. So you know that both bacteria, fungi, and parasites all have ribosomal genes that can be used for detection and differentiation. Virus do not possess these genes, so we could not include viruses on this platform. So the PCR amplicons are sequenced using Illumina sequencing technology, and the output, the reads that we get, are annotated automatically to taxonomic level by a software called Bion, which is a software that was developed for this particular purpose, but which is actually free for use and for download. We also, in this assay, have the option of retrieving FASTA files. So if we want to sort of manually confirm a diagnosis on an organism, we can do that, and we can also make consensus sequences and blast them. And if we want to sort of scrutinize genetic diversity within some of these organisms. So the result of this, the use of this assay, is that we can sort of approach a total detection and differentiation of ribosomal genes in a given sample. So we did a study a couple of years ago where we asked ourselves whether 16S and 18S analysis is a cost-effective method for detection of non-viral pathogens involved in infectious keratitis. And specifically for acanthamoeba, we wanted to identify the specificity and sensitivity of the metabarcoding platform compared with the real-time PCR that we usually use to detect acanthamoeba in corneal scrapings. And also, on a more general basis, we wish to evaluate the clinical applicability of the metabarcoding platform in terms of simultaneous detection of fungi, parasites, and bacteria as causative agents of keratitis. This could be a quite attractive solution because as of now, you have to have a lot of knowledge to sort of find out which organisms you should test for and in which order. But here, this could be sort of like a one-fits-all, or at least a one-fits-most approach that you could use upfront as a primary diagnostic tool. We included 200 DNAs from corneal scrapings, and the number of samples positive for acanthamoeba was 19 by the metabarcoding platform, and 20 plus 2 weekly positives by real-time PCR. So the sensitivity of the metabarcoding platform relative to real-time PCR was 95% or 86% if you include the 2 weekly positive ones, and the specificity was 100%. And when we looked at the reads that we got from our Illumina sequencing of the PCR products, we saw that the average total sequence read per sample was about 110,000 for all 4 prime methods combined, that is for bacteria, fungi, and parasites. And I've listed some of the agents potentially involved in the development of infectious keratitis in these patients here. And also you can see that we had to sort of establish a cutoff for when an organism could be thought of as clinically relevant, because this is far from straightforward, and especially if you don't have any reference data, if you don't have access to the local clinical microbiology laboratories where you have all the data on microscopy and culture, it was quite difficult for us to develop these cutoffs, these thresholds. So we took a quite conservative approach to identifying these. But you can see that we actually found something that could explain the infectious keratitis in 84 of the 200 samples. So that was quite a lot. And for bacterium such as Pseudomonas aeruginosa, we found up to 94,924 reads, for Moraxella up to 19,643 reads. For the mold Fusarium, we found up to 86,399 reads for Acinetobacter baumannii, 68,808, and Saccharomyces cerevisiae we found 74,000 up to 74,376 reads. So we are actually still trying to obtain permission to use data from MIBA, this national clinical microbiology database, to compare our findings with the local findings from the routine diagnostic tests used locally. And also we are trying to actually carry out a prospective study for confirming the applicability of this method. The very interesting thing is that we, as I mentioned in the beginning, we are able to extract FASTA files. We cannot see the sequence chromatogram, so we cannot do any FRET score determination or evaluation, but we can retrieve the FASTA files. So we can generate consensus sequences, which we can query against the NCBI GenBank database, and we can identify, for instance, the genotypes of Acanthamoeba involved in these samples, in these infections. And so we published this in our publication, where we sort of reviewed what other colleagues have been finding in corneal scrapings mainly. And our data is down here. That's the Holmgard publication from 2020. And you can see, as you know or might know, we don't really use species names for Acanthamoeba. We use the T number system. So we call them genotypes, but they are arguably separate species. But they are referred to as genotypes. And the genotype T4 is the genotype so far most commonly involved in infectious keratitis, and our data confirmed this. 19 out of the 21 that we had sequence data for were T4. Only 2 samples were not T4 but T6. This finding was actually quite conspicuous, because T6 has not really previously been identified in cases of infectious keratitis. I think I'll skip this one. So these data were published, our preliminary observations were published earlier this year in Journal of Clinical Microbiology, and I hope that you would be interested in looking up this paper, or you can email me at run@ssi.dk, and I'll provide you with a copy. So, of course, this study had some limitations. It was a retrospective study, and although all clinical microbiology data in Denmark are compiled in this national database, we did not have permission at the time to look up relevant results from local clinical microbiology departments, including data on culture and microscopy that could have served to evaluate the method, the metabarcoding method in greater detail. And also we had to use arbitrary cutoffs that were not informed by data from local clinical microbiology labs. And we're currently actually trying to get money for a prospective study involving a PhD student together with colleagues in Aarhus. And this study could potentially evaluate the diagnostic value of using the microbiome platform as a first-line screening tool for non-viral pathogens as causes of infectious keratitis. So I want to thank you all for listening, and I want to thank the organizers again for including me in this series, and I'm happy to take questions, and you're always welcome to email me at run@ssi.dk. So thank you very much for listening.